This program is a presentation of UCTV for educational and non-commercial use only.
Because our theme is, to some extent, the transitions to industry, I thought I'd describe how, in one set of instances, we're trying to use this interface between bio and nanotechnology, learn lessons from nature that might be translated into economic advantage. And as you'll see, it's a terrific team of graduate students, undergraduates, and postdocs that have done the work that I'll be privileged to talk about today. We take our inspiration from the remarkable nanostructures made by living organisms from the element silicon. And many creatures produce structures of silica, that's silicon and oxygen, glass, including some sponges. They produce, these are electron micrographs of some of the structural and defensive structures made by sponges. This is a interlocking conformal skeletal network. It's quite remarkable how the cells produce this material. And this is a photograph of the skeleton of a deep sea sponge. We've removed the outer tissue, and as you can see in the close-up, it looks as if this material is made out of woven glass. In fact, it's not really woven at all. Our analyses showed that individual cells produce relatively simple, small subunits of the glass, which they position and cement together to make this hierarchically structured material. There's several remarkable things here. First, the material it's crush resistant. It's lightweight and remarkably strong. I don't have time to talk about the architectural principles that make that possible, but it's also quite amazing. Biology, living organisms making glass at low temperatures, near neutral pH, no caustic chemicals within living cells. Whereas in technology, we need high temperatures and very caustic chemicals to make glass and other forms of silica. How does biology do it? In order to answer that question, we turn to a simple sponge that produces simple glass needles as the skeletal element. It's a sponge locally available right offshore from our lab. It fits in the palm of your hand. If you dissolve the tissue, you're left with a handful of fiberglass needles that are shown here. They're 2 millimeters in length, 30 microns in diameter, and this turned out to be a remarkably tractable system that made it possible for us, using biotechnology, to learn first how the silica is made. And from this, we discovered the applications of the underlying principles extend far beyond the element silicon to many, many metals and metal oxides. Here's a photograph of the sponge cut in half. You can see these bundles of fiberglass needles, kind of whitish bundles that form a rudimentary skeleton. When dispersed, the needles look like this. In high magnification, you can see that there's a protein filament that runs right down the center of each one of these needles, and that protein is, of course, the key. I don't have time to describe all the work that was involved, but we found that by dissolving the glass, we can purify these protein filaments. So you're left with a very pure preparation of spaghetti-like protein filaments that are 2 millimeters in length and about a 1 micron in cross-section. And they're composed of only 3 very, very similar protein subunits. And when we cloned the DNA that codes for these proteins and determined the sequence, we found, to our surprise, we know that protein. In fact, that protein is, that family of 3 closely related proteins are highly homologous to a well-known family of enzymes, catalyst proteins that are involved in many areas of intermediary metabolism. They are hydrolytic enzymes. Some of them are known as proteases, others as esterases, lipases. They catalyze hydrolysis of biochemical precursors. And this led to the suggestion for the first time in the 200 years of biomineralization studies that perhaps the protein inside the mineral is actually functioning catalytically as an enzyme. And in fact, it turns out that it's functioning as a catalyst, an enzyme, to make the glass and simultaneously serve as a template guiding the deposition on the protein filament to make this filament-structured glass needle. Because we cloned the DNA coding for the protein, we were able to express it in bacteria and test this hypothesis that I just described, that it is responsible as both a catalyst and a template. And we found that, sure enough, the protein alone is capable of catalytically producing glass at low temperature, at neutral pH, from molecular precursors, which are first hydrolyzed prior to the polycondensation to make up the silicon-oxygen network, the silica. Because we have the DNA cloned and we know the structure at the correspondence between the nucleotide sequence at every point and the corresponding amino acid sequence, we could test our hypothesis that the same amino acids that are known to form the catalytic center in the other hydrolases might actually be involved in the catalytic mechanism in the formation of glass. And sure enough, when we changed the DNA to produce a variant of the protein in which the nucleophilic serine hydroxyl is replaced with a methyl group, we knock out 90% of the catalytic activity. If we change the imidazole, which we postulate is hydrogen bonding to that nucleophilic oxygen and increasing its nucleophilicity, facilitating its attack on the silicon, when we place the imidazole with the methyl group, we knock out 90% of the activity. If we replace both of these catalytic amino acids together, we knock out 95% of the activity. If we take the imidazole replaced protein, we find we can rescue the activity by providing imidazole in solution. These and a variety of other genetic engineering and molecular modeling experiments and analyses have given us a good understanding of how these 3 amino acids, the 2 that I mentioned and one other that assist in the catalysis, participate in catalyzing the hydrolysis of molecular precursors like tetraethyl orthosilicate at neutral pH to form the active intermediates, which then condense to form the silica or glass network. We found beyond this that, silicon is a metalloid. It's metal-like in its chemistry. And in fact, we found that the way that biology is dealing with silicon is similar to the way in which it interacts with many metals. And so using that as a clue and what we understood from the genetic engineering experiments, we realized that we could probably make the same silicatine protein and structurally direct the evolution of structures of metal oxide semiconductors like gallium oxide, titanium dioxide at neutral pH and low temperature. Conditions very different from those that the semiconductor industry is presently using. In fact, we've used, hello, Jim? Okay. We have to reboot our projector. So as I'll show you in just a second, we found that we could compare and overlay the crystallographic structure of the metal oxide semiconductors that we're making with this enzyme on the molecular structure, there we go, of the protein. This is a little section of the protein filament to identify not only the catalytic parts of the protein, but those that are involved in the templating, the control of the nanostructure of the protein. Here's the crystallographic structure that we determined by electron diffraction of the gallium oxide that we made at low temperature and neutral pH with this enzyme. And here, the underlying structure of the enzyme that serves as both the catalyst and a template. In the green are the amino acid functionalities that serve as the catalyst. And we've been validating our hypothesis that these rows of hydroxyls form an extended array that stabilize the emergent nanostructures of the crystalline semiconductors. This was very exciting to us. The catalyst we discovered is the template. That's a design paradigm that's completely different from anything I know in biochemistry for the carbon-based world of metabolic products. And it's an interesting design principle. We thought perhaps because we're able to make nanostructured semiconductors at low temperature with, in some cases, fairly unique structures, our colleagues in electrical and computer engineering might be able to use these to advantage. But when we asked them, could they do so, they said, we don't want to use proteins, we don't want to use biomimetics. Well, this drove us to first understand more deeply and validate the underlying mechanism of the reaction that we identified by genetic engineering, as I mentioned, and some other, let's say, high-resolution analyses. And so working with Tim Deming, who at the time was at UCSB, we made biomimetic, inexpensive counterparts simply by making block copolypeptides of the amino acids that we identified by genetic engineering as required for the catalysis. And when we made these synthetic block copolypeptides, they self-assemble because they're amphiphilic, and sure enough, they catalyze the formation of silica from the molecular precursor at low temperature and neutral pH, and they also serve as templates. If we allow these to concatenate so they make long templates, they template the formation of long fibrillar bundles of silica. If we block the concatenation, they would only make small glassy beads. So we began to get confidence that we were understanding the molecular mechanisms in detail. We took this a step further by taking the functional groups, the nucleophilic hydroxyl and the imidazole that we've identified as required for catalysis, coupled these through alkane thiols to gold nanoparticles, and asked, will the interface form the catalytic unit, just as it appears to do in the biological protein? And sure enough, here's the magic moment illustrated by Peter Allen. When those two functionalities come together, it's the interface that forms the catalyst. We found in these experiments that neither alone is catalytically active, just as in the case of the genetic engineering, or if we substitute one or the other with methyl groups, they also, the combination is also inactive, just as in the genetic engineering. We took this a step further, putting the alkane thiols on a gold surface, or making the corresponding planar corresponding self-assembled monolayers with the catalytic units here on silicon wafers, mimicking the enzyme. Again, we found that we could use this biomimetic self-assembled monolayer to write nanostructured patterns of semiconductors, metal oxide semiconductors, on a silicon chip at low temperature. Here, Peter's drawing again of that interface is shown in these experiments too to be the catalytic unit, and we were able to prove with these experiments that it is in fact the hydroxyl that is the templating unit. We were excited because this is a low temperature biocatalytic counterpart to metal oxide chemical vapor deposition, the high-tech method that Intel and others use to make semiconductors. But our colleagues in engineering asked, can we get rid of the carbon completely and find a way to make pure semiconductors without any organics? So we had to take a step back and translate from what we learned from the biomolecular mechanism into the world of pure chemistry and what I'd call hard engineering, with no biochemicals, no biomimetics. And so what are those lessons? We learned from this system that first, the enzyme is working catalytically relatively slowly, actually 5 orders of magnitude more slowly than the corresponding enzymes work in the world of carbon. That's important because this slow kinetic control bleeds in the reactants that are going to be the precursors for the final polycondensation and make the semiconductors slowly enough to defeat the thermodynamic default that you get in bulk precipitation or from high temperature methods, allowing the weak cooperative interactions of the nascent nanocrystallites with the underlying molecular template to determine the final emergent structure, determining vectorially the control of the emergent crystal structure, regulating the polymorph, that's the kind of atomic lattice, its orientation and connectivity. Could we do this synthetically? Well, it's easy enough to slow down the rates of reaction, lots of ways. Most people in this game, as you know, have been using genetically engineered phage or self-assembled monolayers or genetically engineered bacterial cells to provide the template. Can you get rid of the template completely? We realized that a template providing this vectorial control is simply a means of anisotropically structuring the reaction environment. When you start thinking that way, you begin to realize that there are ways to do this without organics. One simple way that we have first used is to use vapor diffusion of a low concentration of the catalyst, a catalyst of hydrolysis of molecular precursors. These can be, we've shown, either alkaline or acid or even water vapor, diffusing through a solution of a molecular precursor that must be hydrolyzed prior to polycondensation to form the metal oxide network. In this case, we're using a solution of cobalt chloride. It's actually a molecular complex, the aqua or hydrated complex, which must be hydrolyzed usually either by acid or alkali. And in this case, we're using a low concentration of ammonia to diffuse through the meniscus of this molecular precursor solution, creating a vectorial gradient in space and time that governs the emergent crystal growth. The result in this and many other cases is the formation of a nanostructured thin film that forms first by island nucleation on the surface of the meniscus, then consolidates, and then through homoepitaxial nucleation provides the direction for further nanostructured growth, which I'll show you in a moment. Thus, we have slow catalytic hydrolysis of a molecular precursor providing the kinetic control of crystal growth with no organics. Nanostructured thin films, in this case it's cobalt hydroxide and many others, that form first, forming that backplane, as I mentioned, through consolidation of island nuclei, and then homoepitaxial nucleation, allowing further growth into the solution of the molecular precursor of highly crystalline nanostructures, which, depending on the particular material, may be either flaky or needle-like. These materials seem to offer a number of advantages, especially for energy applications, because of the very high surface area and their very high crystallinity, as shown by the electron diffraction. We found that we can make many novel materials and nanostructured materials this way in morphologies that cannot be made at high temperature. And as a result, we've discovered in some cases very useful electronic or optoelectronic properties. The method is relatively low cost and provides a generic method that we have used now for making more than 50 different nanostructured thin films or nanoparticles of metal oxides, hydroxides, or phosphates, and converting many of these post-synthesis through metathesis reactions to structure-conserving nitrides or sulfides. Again, families of valuable semiconductors. Let me illustrate this with just one example of the kinetic control provided by this low-temperature, biologically inspired method that provides a unique kinetic control of the atomic structure and electronic properties. It's really an unrealized power of this approach to control nanostructures. Am I getting away from the mic? All right. So in this case, James Nielsen and our group has used this method to control the proportion of tetrahedrally coordinated cobalt atoms in cobalt hydroxide thin films, such as I showed in that electron micrograph, ranging from either the material which is 100% octahedral to about 40% tetrahedral. Interestingly, it is this material which is strongly absorbent in the visible light. It shows very strong photovoltaic activity, magnetic activity, and even photomagnetic activity. Some of the photovoltaic behaviors shown here. We've produced the first working examples of biologically inspired photovoltaic solar cells with no organic templating whatsoever. This kinetically controlled synthesis, we believe, offers a unique opportunity to optimize structures like this responsible for these important behaviors, and thus to explore, among other things, a revolutionary new pathway to energy conversion, photomagnetism, a way of translating the sun's energy into electricity without suffering the losses that limit all photovoltaic applications because of the losses of the photoelectrons at all the junctions that are involved. Here, in photomagnetism, the aim is to withdraw the energy inductively into an AC coil, the preferred mode of electrical transmission. One of the great advantages of this method is that if you start with a bimetallic precursor, where the geometry at the atomic scale is built into the precursor, you can preserve that geometry using this gentle biologically inspired catalytic method, incorporating these units, preferentially hydrolyzing off the alkoxide groups in this case, or the chelates, depending on what the structure of that precursor is. In this case, to produce the smallest, most uniform nanocrystalline barium titanate yet achieved without any phase segregation. Usually the high temperature methods for production of these perovskite-like materials results in some production of the desired material, but contaminated with phase segregation, that is, in which you get an island of one of the metal oxides surrounded by a sea of the other. Here, this material, 6 nanometers, remarkably monodispersed. It's plus or minus about 2 nanometers, and ultra pure with exceptional crystallinity and purity. Here you see in a high-resolution transmission electron micrograph the rows of atoms of this perovskite. We're pursuing a number of potential applications. I'll describe just a couple of them. Here in high resolution, each one of those nanoparticles is a single crystal. We found that we can readily disperse these to make thin films of these materials for a number of applications. We've found that the method is scalable. That is, we started with small dishes of reactants, with the vapor diffusion from one to the other, producing just a few milligrams. Recently, Thea Buldelli and Christian Nies in our group have been able to scale this up using vapor delivery of the catalyst into a stirred solution, with producing routinely now 100-gram batches. Again, the material highly crystalline, 6 or 7 nanometers, ultra pure, and quantitative yield, 100% conversion. They're working on the construction of a reactor that's about the size of a person that is designed to produce kilograms, and recently have found that spray dispersion in fine droplets combines the advantages of kinetic control with very high throughput, again with quantitative yield and preservation of these properties. As a result, in a nutshell, what we've discovered from the biological mechanism of the nanofabrication of silica in sponges has been translated into a robust, low-cost, low-temperature method for the synthesis of a wide range of nanostructured thin films and nanoparticles. It's a generic method producing many novel structures with novel activities. And so with industry partners, we're pursuing a number of potential applications ranging from lightweight high-power batteries for high-power hybrid electric vehicles and medical implants, solar energy at lower cost, uncooled infrared for medical diagnostics and other applications, and 3D medical ultrasound. And let me just tell you in the few remaining minutes of a few of those of our recent progress. In one, Dr. Hongli Zhang took our method in a completely new direction to produce novel tin-infiltrated graphite anodes for better lithium-ion batteries. They perform anything that's out there so far. Present anode material in lithium-ion batteries is graphite. The electrochemical capacity of graphite is low. The metals have much higher electrochemical capacity, but each time the lithium ions enter the metal to form an alloy, the metal expands by a factor of 3. When the lithium ion leaves the metal in each charge-discharge cycle, the metal shrinks. Multiple cycles of charge-discharge produce disintegration of the metal, leading to its loss of connectivity to the electrodes. What Hongli did is he used our vapor diffusion catalytic synthesis method to grow the tin in situ inside the pores of a compliant graphite matrix using microparticles of graphite. He infused these in a simple low-cost solution of tin chloride. Vapor diffusion catalytic synthesis converted the precursor to nanoparticles of tin oxide in situ, and then slight heating allowed carbothermal reduction to convert the material to highly crystalline nanoparticles of tin metal, preserving the crystallinity of the graphite. The result is that you have the tin metal with its higher electrochemical capacity in a compliant, resilient graphite matrix that can breathe and accommodate the expansion and contraction of the nanoparticles of tin. The result, as you see here, is a 30% higher electrochemical capacity than the present commercial graphite with rock-stable stability. And if you try to do this any other way, slowing down the chemical reaction with conventional means, you don't get there. You produce tin nanoparticles on the outside, you get the initial high capacity, but because the particles are only formed on the outside of the graphite, they eventually swell and shrink and disintegrate and fall off of the graphite. So this is, we're very excited, it's going to appear soon in Journal Materials Chemistry, and we're developing this with a new small company, LifeCell. This graph shows the relative performances of different energy sources, lithium primary batteries, lithium-ion batteries, comparing the specific energy and specific power, and you see that this material significantly outperforms the conventional. I don't know, Frank, are you my chair? Do we have 2 minutes? 5? Are we okay? Then we're also using the ability to produce the nanoparticles of barium titanate to make an internal protectant to make fireproof and explosion-proof lithium-ion batteries. When you dope the barium titanate and then sinter it, you make a fine-grained nanoparticle nanocrystalline ceramic. It is, in fact, the finest grained nanocrystalline ceramic of this material yet obtained, and that's because we started with such small nanoparticles to begin with. The doping makes it possible to make the barium titanate serve as a conductor normally at low temperature, but then, unique property of this material, it exhibits a 10,000-fold increase in resistivity, a positive thermal coefficient of resistivity when heated above the Curie temperature. It's transitioning here from a conductor to an insulator. And of course, we're thinking of using that as a thin film of the ceramic on the electrodes to provide protection against thermal runaway in response to the heating of from a short circuit, rather than igniting the flammable electrolyte. The barium titanate PTCR protectant would cut out the short circuit. If I have 2 minutes, let me tell you that we've recently found that this approach of looking at high-performance systems in biology and trying to translate into new value-added applications in the synthetic world is not confined to inorganic materials. For example, in the world of organics, talking about biophotonics. Cephalopods, octopi, squid, and and cuttlefish are the masters of dynamically tunable biophotonics. A second after my friend Roger and collaborator Roger Hanlon took this photograph of a Caribbean octopus attached to a rock, as he approached it, the animal changed its appearance. He's still there. You can see the eye. It's changed his color, his texture, and his reflectivity, all with neuronally activated changes in different layers of the skin. We've been working on the layer in the skin that is tunable in terms of its reflectance and color. There's a layer of cells which Daniel DiMartini and Alison Sweeney have been working on in our group, and Andrea Tao has worked on, that dynamically can tune to show any color, as you see here. This is the same species of cuttlefish in two different displays. This is Stay Away, and this is Come Get Me for Mating. And I know you can see that was attractive. The way in which this works is inside those cells, there are organic structures, lamellar structures, membrane-enclosed structures, rich in a unique family of proteins called reflectins that form Bragg reflectors. These are tunable Bragg reflectors that progressively change in response to neuronal activation. The assembly of the proteins drives, we found, the densification, increasing the refractive index, and the thickness and spacing of these reflectors also change. Here, in response to a pulse of the neurotransmitter acetylcholine, you see the progressive wave of color changing from red, yellow, all the way through blue in these cells. And in a nutshell, we discovered the molecular cascade that has been referred to in the symposium before, in which the neurotransmitter activates a signal transduction cascade, culminating, let me say, in the activation of a protein kinase that adds phosphate groups to these reflectins. And we've also been working to understand how the changes in charge on the reflectin proteins overcome the initial high positive charge to allow condensation of the proteins in these lamellar organelles to drive the increase in refractive index and the change in thickness. Working with our colleagues at Raytheon, Amanda Holt in our group has translated this information into an electrically switchable, polymer-based shutter for infrared detectors. Here, when low voltage is applied, the polymer thin film converts from being transmissive in the infrared to opaque. The next steps in this project are to add electrostrictively deformable photonic crystals for a high-gain tunable multispectral filter. In other words, to tune through the colors in the infrared to make this a lightweight multispectral filter, and then also taking advantage of the shutter to develop an electronically coded, pixelated set of apertures for image formation by information synthesis without a lens.
The main focus of our research lab is to look at nanofluidic technology, and we do a lot more things other than biology, but I'll focus on why we're doing nanofluidics for biology. But first, I want to back up and actually answer the question, what is nanofluidics? My definition of nanofluidics is not the normal one, which you would think of as anything that flows, fluid that flows in nanoscale channels. I'm actually really interested in looking at things where the physics are different than microscale or macroscale. So, and the physics are different because it's in the nanoscale. So there are many things that are going to be occurring where the physics I'll be explaining here today, where the sizes are actually almost micron size, but you still get the same physics. And there's other physics where, or the scale is nanometer, but if it's following continuum theory or macro scale theory, I don't really consider it nanofluidics. So that's sort of a basis of a starting point. Now, why nanofluidics for biology? Well, there's a lot of advantages. Some of it has been gone over in the last two talks, but it's a very small sample volume, smaller sample volume than anything else. We need less than a nanoliter of volume in our samples. We can do everything in free solution, and I'll talk about that later. This is one of the advantages of the nanoscale. Very small diffusion times. Molecules are going to see each other much, much quicker because it's in a confined area. We have very high sensitivity. We'll talk about that later. We can enhance reaction kinetics, again, because of small diffusion times. But the big deal is that we can analyze, separate, manipulate, and interrogate individual molecules at the nanoscale instead of having an aggregate of molecules. And there's many bioanalytical applications to this, including DNA fingerprinting, virus detection, molecular recognition, different protein isoform identification, oxidation state mappings, and I'll talk about some of these during the talk. But the real overarching goal is that we want to develop a new technology for biomolecular analysis that surpasses performance of traditional tools. Not just faster, better, cheaper, but can also do things that no other tool can do. So I'm starting, unlike the other two spots, with my vision. The vision is to eventually use nanofluidic technology as a handheld device. The other great thing about nanofluidics is you can make it the way computer chips are made in very small devices, and it can be all integrated in a handheld device. So we have a vision that you can take any sort of bio sample, urine, blood, saliva, whatever, and put it into a handheld device. Now we're working with people at Lawrence Livermore National Labs in Boston University to talk about sample prep, to get from the sample prep part to the samples that we need, which would be, for example, DNA. So this is a sample prep part, and this is the chips that we make in our lab. And you can see there's a hybridization chip here. We put the DNA on one side. Right now we conjugate it with a fluorescein molecule. This is one of the reasons I want to talk to the other speaker. This might work well if it's a conjugated polymer, but we conjugate it with a fluorescent tag, a single stranded tag of DNA. Because it's flowing down in nanoscale dimensions, it actually hybridizes very quickly, within milliseconds, and I'll show you some examples of that later. We drive the fluid from top to bottom. We inject some of the fluid by changing the electric fields from left to right. And what happens is we have a laser downstream that can measure the peaks as they come by. The DNA all separates from each other, and it develops a unique signature, and I'll talk about that later too. And we can get this signature, it's called an electrophoregram, and that will be our signature that can help analyze some sort of diagnostic or disease or something. So that's sort of our ultimate goal. Before you can understand how this whole ultimate goal becomes reality, I'm going to give you one background slide on general electrokinetics, how we move fluids at the nanoscale. It turns out that in fluid dynamics, microscale, nanoscale, pressure-driven flow is hard because you need a lot of pressure to drive through a tiny conduit. So we use electrokinetic flow. We drive flows with electric fields. How does that work? Well, every wall in a channel is charged. Almost every surface is charged. We generally use silica, glass. Glass is negatively charged. So when you put a fluid, an electrolyte solution, into that channel, you get a region of ions that are positively charged, that are attracted towards the wall. Now, if the microchannel was the size of this room, the size of this electric double layer, the region of positive charge, would be the size of the paint on the walls. It's very, very thin, right? Maybe a nanometer, 10 nanometers big for a microchannel. But it still allows you to apply an electric field from left to right, and these positive ions, the paint on the walls, will start moving towards the negative electrode. And that will drag all the rest of the fluid along with it, causing a fluid movement called electroosmosis. The individual movement of ions within the fluid is called electrophoresis. And what we can do is, this is in a microchannel. This is like a 20 by 30 micron channel. It's a cross channel. We can drive fluorescently tagged molecules from the top channel to the bottom channel. So we have the sample well up top. We're driving it from top to bottom. And then we inject a little bit of the fluid from left to right. And what you're seeing is you see two peaks separate from each other, right? And that's because in this sample, we actually have two different fluorescent molecules. One's fluorescein, one's BODP. And because they have different electrophoretic mobilities. Now, that's just a property of the of the fluorescent molecule, just like diffusivity or charge or size. If they have different electrophoretic mobilities, they separate. And so if you have a camera downstream, you can actually watch the peaks as they go by the detector and construct an electrophoregram. And the reason I'm going over this slow is because I'll be showing you a lot of electrophoregrams during the rest of the talk. Now the big thing is this is all well known. It's been done for 20 years. It's been done in microchannels in the last 10 years or so. People use it all the time. But what we're doing in our lab is doing it at the nanoscale. Now what does that mean? It means that we're shrinking the size of the channel from the size of this room to the size of the paint on the wall, right? So why is that so great? Well, in a microchannel, you have a very thin electric doubler. Say I happen to have two ions moving at the same exact speed. They have the same electrophoretic mobility. They won't separate from each other in a microchannel. But in a nanochannel, all of a sudden, your channel's filled with charge. And your positive 2 ion is going to be attracted more towards the wall than the negative ion. The fact that you have charge everywhere also makes for a non-uniform velocity distribution. You solve Navier-Stokes equation with this added body force term, and you get a non-uniform velocity distribution. What does that mean? It means that you get coupling between the slower moving ion close to the wall and the fact that it's slow near the wall. So the larger your charge is, the slower it's going to go. So if you have this two charges with the same exact electrophoretic mobility, they wouldn't separate in a microchannel. Because of this unique transverse electric field and non-uniform velocity profile, they separate in a nanochannel. So this allows you to separate, do something that you could not do in a microscale, something that you just can't do otherwise, and no other technology can do this, but the nanoscale allows you to do this for small ions. So we've shown this theoretically. This is a student, David Boy, in our lab, worked out this simulation theoretically, just solving the Navier-Stokes equation. And what you're seeing is a channel that extends all the way this way to all the way that way, and one with a more negative species that is traveling down the channel, and one with a less negative species that's traveling down the channel. These are small ions. And what you see is in the channel, we're looking at the cross-section area height, which is about 100 nanometers. What you see is that the more negative is, the closer to the center of the channel it is, if the wall is negatively charged. And therefore, after the amount of time steps that you just saw in the movie, the more negative analyte is going to be a little bit further up than the less negative analyte. So we can separate things even though they have these particular have the same mobility. So we did this theoretically. We also did this experimentally. What you see up top here is a 1 micron wide by 2 micron deep, and then 1 micron wide by 40 nanometers deep. You're seeing the same sort of separation I showed you a couple times before. We flow from top to bottom and then go from left to right. And what you see is the same fluorescein and bodopy, and you see different separation dynamics. So just to look at this a little bit in more detail, this is the first screen. This is near the end. Fluorescein is a -2 charge analyte. Bodopy, this particular one we bought, was -1 charge. So what we see now is in the nanochannel, the fluorescein has caught up to the bodopy. We kept everything else the same here, the electroosmotic flow the same. So it turns out that a -2 charge is moving faster in a nanochannel than a microchannel. And so we prove that, hey, look, small ions exhibit all this different behavior. But what happens if we actually use biomolecules, right? Because everything I said was small ions, everything's continuum theory. It's really easy to do mathematical relations towards this. But biomolecules introduce a lot more complexity. We have hydrodynamic drag on the molecule. We have Brownian motion on the molecule. We have adsorption kinetics with the walls, axial and transverse electromigration, electrostatic interactions with the wall, and everything will hopefully work with us to try to get better separation techniques. But the physics involved are very complicated. Before we decided to delve into the physics, we wanted to see if it works. Hey, can we actually separate DNA in a nanochannel better than in a microchannel? And most of you know that in a microchannel, in free solution, DNA doesn't separate because they have the same mass to charge ratio. So what you're seeing is an electrophoregram of 5 different DNA molecules, and all end tagged with fluorescein in a channel. It's in 10 millimole borate buffer. It's 1 nanometer electric double layer thickness, very thin double layer. And what you see is after 70 seconds, you see a peak of fluorescein, and you see 5 peaks of all the DNA in 1 peak. 1 base pair, or UTP, 10 base pair, 25, 50, and 100, all in 1 peak. It's not separated. If I run the exact same experiment, but I shrink the channel down, so it's 100 nanometers, the exact same experiment, and I just superimposed it, you see 5 distinct peaks of DNA. So that was a really exciting result. We actually saw a separation that you could not see in the microscale. After the same amount of time, we saw separation in the nanochannel, which you don't see in the microscale. So that's great. We found a new way to separate DNA. The question is, which peak is which, right? And if you think carefully, we would think that the 100 base pair DNA should come out first because it's most negative. But it turns out what we did was this black electrophoregram down here is the same exact one you saw in the last slide. And then we took the same exact solution and added 5 times higher concentration of 100 base pair. We figured whatever peak came out first is the 100 base pair. Then we did it with 50. Then we did it 25, 10, 1, and fluorescein to figure out where the peaks, what order the peaks came out in. And in reality, they actually came out in the opposite order of what I just told you with small ions. It turned out that the 100 base pair peak came out last. And since it's most negatively charged, we think it would come out first. Right? So that didn't really make sense to us. Then we went back and looked at the nanochannel and we said, wait a minute, the double layer thickness is only 3 nanometers in a 100 nanometer channel. So maybe it's not the size of the paint, but maybe it's the size of a couple books or something. It's not that much of the channel. What if we make this electro double layer thicker? Maybe that will do something. And it turns out that it did. It actually switched the order around. 150 base pair did come out before the 10 and 25 base pair. But it was still very, very confusing to us. And so we had a choice here. Whether to keep going with the DNA separations and try to show exactly what we could do, or to step back and do more fundamental studies. And what we decided to do in our lab is do both. So for applied studies, we realized, hey, the nanochannel environment allows for unique opportunities for separation, analysis, and detection. So we should go forward and see how good this technology actually is. So we decided to start with oligonucleotide hybridization and separation. Can we actually do what I said the ultimate goal was to do? Can we take a single strand of DNA, hybridize it, and separate it all in 30 seconds in a handheld device? And I'll show you that work next. Another thing we started to do is we coupled with a professor in physics, Deborah Feigenson, who's been working on silver DNA nanoclusters. And they don't know much about those silver DNA nanoclusters, and we said, hey, maybe we can use our technology to help characterize those molecules. We also have been working in the Bren School with titanium dioxide separations and characterizations, and we've done some initial work with well-characterized proteins. And these are all just applied. We want to see how far we can go with the technology. But we also want to take a step back and look at the separation mechanisms and figure out exactly what's going on. And to that end, we've developed total internal reflectance microscopy studies of particles. We've determined ways to pre-concentrate samples so that we can really detect low concentration samples. And we have a study where we do well-characterized particle separation, spherical particles, just trying to figure out what's going on. So depending on time, I'd like to try to hit a couple slides on each of these. The first one, the most impressive, is DNA hybridization and separation. So what we're doing here is exactly what you saw in the movie. We have a single strand of DNA on one side, another single strand of DNA that's an exact complement with a fluorophore on the other side. We drive it from top to bottom, we inject it from left to right, and we have a detection point where we see peaks come out of the fluorescent tags. And so what you see here is the same thing, single strand DNA and a marker, and we're looking at the electrophoregram. What we do is we see different colors here, 0 seconds to 600 seconds. That corresponds to the time we let it hybridize for. So right away when we do separations, this is a fluorescein peak, by the way, we see a lot of single-strand DNA and not that much hybridized DNA. After 10 minutes, it's all hybridized. So we can actually get, on top of separating these, we can get hybridization kinetics, right? And in fact, we can use hybridization kinetics to, in 70 seconds, look at the difference between a perfect complement and a single nucleotide polymorphism. So not only can we separate DNA from one another, an SNP and complement, which you can see is a little bit different, the red and blue. After long times, we can definitely separate them. What we can do is take a single-strand DNA and then take one that's one base pair mismatch, and run it through this sample. And depending on how much is hybridized, we can figure out whether it's actually a perfect complement or a single nucleotide polymorphism, which I think is pretty impressive. So we're working more along those lines as well. Another neat study that we undertook is in collaboration with Deborah Feigenson in the physics department. And she and Professor Beth Gwynn have been looking at silver DNA nanoclusters. Now these are silver atoms that can be stabilized by hairpin DNAs. Okay? So single-strand DNAs. And the hairpins that they've been choosing for this is cytosine. 12 cytosine, 11 cytosine, 10, 9, 8 cytosine hairpin loops. And what they see is, depending on how many cytosines they use to stabilize the silver atoms, you get different colors of fluorescence. So it's pretty neat. They can use this as a fluorophore label. It's just yet another labeling technique. It can be used for biomolecular labeling, for DNA scaffolding, et cetera. And the fluorescence can be tuned by DNA sequence. However, not much work has been done to really determine what's going on, what the structure is, what's the size, what's the charge, what's the mobility, what's the diffusivity. And so we figured that we could use our separation technology to characterize the diffusivity, mobility, and charge of the silver DNA nanoclusters. Well, how do we do that? This is work again done by Till Drehorst in our lab. You see right here, we inject a sample down in the channel, and then we stop all the flow, and we watch it diffuse. And from that, we can get diffusivity measurements within the channel. We can also do separation experiments in the nanofluidic channel and figure out where, after time, how long it takes for it to elute to a certain point, and from that we can back out mobility from electrophoretic mobility with all our relations of electroosmosis and electrophoresis I said before. And what you see here are all the different mobility data that we have for different silver DNA nanoclusters. And what's pretty interesting is we see that a lot of it's dependent on pH. We show it for different pH. We have remarkable repeatability. All these experiments are about 10 or 12 times, and they're just really in a small band, which is pretty amazing. We can separate different silver DNA nanoclusters very well, which you can't really do with other techniques. And we can point to different silver DNA conformations. Just looking at the mobility and diffusivity, I don't really have enough time to go into the details here, but we can actually have more information about how these hairpins actually form around the silver DNA clusters. So it's pretty neat. Again, we've been able to show that our nanofluidic technology can work towards learning more about physics of biomolecules that no other technology can do right now. We've also been doing titanium dioxide separations. I'm not going to go much into this because I want to talk a little bit about our fundamental studies. But again, we can get 3 different peaks, and actually 4 peaks. It's really hard to see, not very well defined here, of different titanium dioxide clusters that are in samples that we collaborate with Arturo Keller in the Bren School, who I think he gets them straight from water. And so we can help try to determine toxicity levels. Again, a new way to figure out these technologies. Now, in terms of fundamental studies, well, we really, really want to figure out why they're separating the way they're separating. We can learn about small ions. We understand why small ions do what they do. We have great matches with theory and data there. But in terms of fundamental understanding of how particles move, we're not really there yet. We can see great things happen, but we don't know why. So in order to figure out why, we've embarked on a few fundamental studies. The first one is total internal reflectance microscopy. What that is if you have a substrate and you're trying to illuminate it for fluorescence, instead of shining a laser light directly on it, we're shining it at an angle. And we're having it totally internally reflect on that substrate. And what that does is it has an evanescent wave that forms from the wall. The evanescent wave is about 100 nanometers big. People use TIRF all the time for looking at surfaces of cells, for example, because it only illuminates about 100 nanometers up. In our case, our channel is 100 nanometers. This is pretty neat because we can look at dots, these are quantum dots, and the brighter they are, the closer they are to the bottom of the channel. The less bright they are, the further away they are from the bottom of the channel. And we can actually figure out where these quantum dots are transversely in the channel. It's like 1 nanometer resolution confocal. I don't know any other place that can actually get 1 nanometer dimensions, z dimensions, of where the particles are in the channel. And so what the data looks like is just when we take quantum dots in a channel, in a nanochannel, it just looks like this, just a bunch of dots. But what we can do is we can process them. And these are 5 by 10 nanometer ellipsoidal quantum dots with a 17 nanometer carboxyl coating around them, negatively charged. And these are all the details of what sort of experiment we're running. But we can change them to color and then change them to height data. And so these are actually the heights of the quantum dots in the channel. And then we can use this and try to relate this to some of the collaborative work we're doing with some math and computer science professors, Paul Atzberger and Frédéric Giboux, who are trying to theoretically figure out how this works, and hopefully it can match with some of these experiments. Another thing we're trying to do, I heard this common theme in the last 2 talks, is getting pre-concentration. We want to try to get the most signal out of our channel, especially because we're working in the nanofluidic regime. We don't have much concentration there, right? Even something that's nanomolar in microchannel would be something like picomolar in the nanochannel just because of the small volume. So we don't have much sample to deal with. We really want to concentrate it and measure it and try to increase concentration as much as possible. So to do that, we've decided to embark on something called nanoscale field amplified sample stacking. How that works is if you have a channel and you have your sample in a region that's very low conductivity, low concentration buffer, as opposed to the outside of the channel, which is high concentration buffer, if you apply an electric field in the low concentration buffer, to have conservation of current, V equals IR, you're going to have a higher electric field inside this low conductivity channel than you are outside. If you have a higher electric field, your molecules are going to move faster in that region. So if you have any sort of analyte that's charged, it's going to go faster in this region and all stack against the interface. So we can actually get stacking of fluorescent analytes against one interface. And people have shown 1,000 times and million times pre-concentration in microfluidic channels. The problem, though, is in microfluidic channels, you can't actually inject for a long period of time because the difference in conductivities induces a lot of instability and induces a lot of internal pressure gradients. So it's not a very viable technique in microchannels. It's used a lot, but you can't actually inject a whole bunch of sample, which is what we want to do. We want to just get a whole bunch of sample and stack it all up so we can get as much sample as possible in the nanochannel. However, in a nanochannel, the nanoscale dimension, again, because it's confined, suppresses these instabilities and induced pressure gradients. So what you can see is what we're showing you is one peak. So it looks like an electrophoregram, but it's not. It's one peak that we're just moving the stage down and looking at different points in time from 5 to 25 millimeters. And you can see we inject a long plug, and it shortens to the point where it actually gets really, really high before it diffuses down. Whereas in a microchannel, this is actually only 1 micron deep. A bigger microchannel, we can't even run the experiment because of the instabilities. You have a long peak that even just at 3 millimeters is the highest it will ever go, and it just starts diffusing after that. So we can get really good concentration even downstream of the channel where we can detect our sample in nanoscale channels. And although this is not relevant to biomolecules, we use it all the time with biomolecules, so I thought I'd say it here. And this is some work by Jess Sisterich. Finally, one of the fundamental pieces of work we're doing are particle separation experiments. This is because particles are well defined. We can use different channel sizes, micro and nano. We can use 3 different buffer types, just a huge systematic parametric study. Different concentrations, different applied electric fields, different kinds of particles, negative and neutral, different diameters of particles, 50 and 100 nanometers, different places to measure, redundancy of measurements. So far, postdoc Maria Napoli has worked on this, and she's done over 600 experiments just to look at how particles move in these channels and see whether we can make sense out of the particles versus the DNA, because DNA, again, is a lot more complicated than particles. And this is the overview. She hates it when I show this slide because it's all 600 experiments on one slide. But all 600 experiments, acetate, phosphate, borate, 3 different buffers. And the main point of this graph is to see that microchannel and nanochannel behavior is different. In nanochannel, you do get what you're seeing on the y-axis is the electrophoretic, the normalized electrophoretic mobility, the way that these molecules move in a nanochannel is very different than a microchannel. It depends on pH a lot. It also depends on the size of the particle, if it is 50 or 100 nanometers big, and it also depends on the concentration, 10 millimole or 1 millimole. So there's a lot of things involved, whereas in the small ions, it would just be all one graph. Here you can see there's 20 different parameters, and we're still working through this, especially with the help of some computational scientists. So in summary, nanofluidic devices allow for the fact that we can use small sample volumes. We can do everything in free solution. We have very fast diffusion times and very thick electric double layers, which allow for these neat physics to occur. We've been working on fundamental studies to identify what's important, what are the important physics in this scale, including total internal reflectance microscopy to identify transverse particle locations within 1 nanometer. Field amplified sample stacking, the pre-concentration technique. We're doing particle separations. But more importantly, we've showed a whole bunch of applied studies. We've shown, hey, look, we can hybridize and separate this DNA. We can look at silver Dna Nanoclusters and titanium dioxide. We're working with doing some Army collaborations through the ICB to look at proteins and peptide separation. So we're forging ahead with what we can do with this technology, but we haven't. We still need to step back and get a lot of fundamental understanding of it. So I feel like we're really at the brisk of a new technology here, and I really want to thank everyone I've been working with. These are all the collaborators that were involved with the work that I showed, and the people starred are the students that are working on nanofluidics for biology. As I said, our lab also does some really fundamental work in nanofluidics as well as some energy applications, which I'm happy to talk about. I want to thank the funding agencies and all the people in the group. So thank you, and I'm ready for questions.